My name is Anna Hodgson. I'm a member of the BioNerve team. This poster presentation will briefly cover the objective, specifications and requirements, product design, overall schematic, results of a hypoxic concentrated fluid test, and the future work and implementation of this project. The overall objective of the BioNerve project is to design and produce a compartmentalized culture system for inducing hypoxia on the axonal ends of a neuron. The main requirements and specifications include an in vitro model of a system of neurons that can be housed such that the cell bodies and axons can be held at varying oxygen levels. The hypoxic media will need to be transferred to a syringe pump while it remains at less than 2% of oxygen concentration.
Hello, this is Tom Yang. This is the 3D printed 2-chamber system for our project. This is the 3D printed lid for the 2-chamber system. This lid will go on one chamber, leaving the other chamber exposed to the atmosphere. Both designs are made up of chemical-resistant PLA. The lid will have an FDA-approved non-toxic O-ring wrapped around the insertion in this image.
Hello, my name is Ryan Gubert, and I'm going to briefly describe to you the overall design of the experiment. The first step in our experiment is to create the actual hypoxic media. The BioNerve team has made significant progress on this step. After the media is created, it must be transported into a syringe pump, which is how we will control the flow into the system. The media then flows through an oxygen sensor and into only one side of the 3D printed container. Inside the 3D printed container is where the ends of the neuron are separated. After the media leaves the chamber, it simply flows into a waste bin and the sensor readings are recorded on a laptop for analysis. The BioNerve team was able to test creating the hypoxic media by first exposing just water to a vacuum, given that the media is expensive to create. We were able to achieve 6% oxygen concentration. This will further be improved by adding nitrogen into the procedure by alternating between exposing the media to a vacuum and then to the inert gas nitrogen. Further tests on making the media hypoxic will be conducted when the nitrogen tank arrives.
Our future work for next semester focuses on developing our cell culture, which consists of the neurons that our project requires. We will be working with our media to reach an oxygen level of 2% or less. We will also have to test the cultured neurons with our hypoxic media, of course after it has reached hypoxia. We are expecting to see neuron cell degeneration, and we will then be performing live cell imaging using a microscope that will capture pictures every hour.
These are our team's point of references during this project so far.